The human microbiome and its relationship to chronic asthma. A microbiome is defined as a community of various bacteria inhabiting an environment, including the genomes of the bacteria and, in many instances, their host. The human microbiome is comprised of distinct communities of bacteria inhabiting the surfaces of our organ systems. These interact with environments such as the gastrointestinal lining in the stomach and intestines, the skin, and the lungs. Here we will look at how the human microbiome plays a crucial role in health, and specifically how it contributes to the development of asthma. The bacterial cells residing in our body outnumber our own cells by roughly 9 to 1. The human genome comprises approximately 30,000 protein-encoding genes. In contrast, the microbiome may comprise up to 3 million genes in total. With this in mind, we can begin to appreciate just how important the microbiome is to human health. In our gut, bacteria help to break down foods and digestion. Some commensal bacteria ferment complex polysaccharides, and in doing so, produce vitamins and short-chain fatty acids as byproducts. Short-chain fatty acids have anti-inflammatory properties by modifying cytokines produced by immune cells, and they have also been shown to play a role in maintaining the integrity of the epithelial barrier. The regulation of inflammation and immune responses is central to the involvement of the microbiome in asthma pathology. Asthma is a disorder involving reversible bronchoconstriction of the airway, which is attributed to inflammation due to movement of fluid into extracellular spaces, and hypertrophy of the mucosal cells, smooth muscle, and goblet cells. These processes all lead to smooth muscle constriction, a decrease in airway diameter, and therefore an increase in airflow resistance. Inflammation of the airway can be triggered by a large range of environmental factors, some of which involve bacterial infection or bacterial endotoxin, which are often present in house dust, for instance. Bacterial endotoxins can trigger cytokine cascades, resulting in an inflammatory response, triggering asthma. So since we all harbour bacteria in our gut and lungs, and since we're all exposed to these factors, why do some individuals have asthma while others do not? Well, studies show that a large contributing factor are differences in microbiomes of the airway of the gastrointestinal tract between healthy and asthmatic individuals. Many studies have established that the microbiota of the gut impacts the development of inflammatory and allergic diseases such as asthma. A 2012 paper by Russell and others showed that altering the gut microbiota of mice with the antibiotic vancomycin, they were able to induce allergic asthma-like disease. In 2010, Hilty and others found that the bronchial tree was not as sterile as previously thought. Moreover, the bronchial tree of asthmatic individuals more frequently harbored pathogenic Haemophilus species, whereas non-asthmatic individuals were found to have more Bacteroidetes bacteria, which is a commensal strain important in a healthy microbiome. So how is it then that microbiomes affect our immune system? The interaction between commensal bacteria and immune cells is a highly complex one. One example relevant to asthmatic triggers involve our T helper cells. Short-chain fatty acids produced by commensals, as discussed earlier, have been shown to alter hematopoiesis, which is the creation of new blood cells occurring in the bone marrow. Short-chain fatty acids cause an increase in macrophage and dendritic cells, thus increasing phagocytic capacity at the lung epithelium. Furthermore, short-chain fatty acids impair the promotion of T helper type 2 cells. High T helper 2 to T helper 1 ratios are correlated with asthma development. Current available treatments for asthma, such as the inhaled corticosteroids found in asthma puffers, serve to relieve the symptoms of asthma by relaxing the smooth muscle of the airway. Although the field is still emerging, modifications to the microbiome may serve to prevent or possibly even cure some patients of asthma. Some examples of possible treatments are old treatment with lactobacillus species, which, according to this study, has the ability to attenuate major characteristics of the asthmatic response in mouse models of allergic airway inflammation. T cells also play a major role here. Another possible treatment are changes in diet and nutrition. A study involving the feeding of diet high in fermentable fiber content altered the ratio of gut Firmicutes to Bacteroidetes species, increasing the levels of circulating short-chain fatty acids and, through their alteration of dendritic cell capacity, to promote Th2 cell effector function, protected against allergic inflammation of the lung. One possible disadvantage here is the little research conducted into microbiome-related treatment, as it is still an emerging field of study. In conclusion, microbiomes play a large role in human health, and in particular the prevalence and effects of asthma. Treatments involving microbiomes, such as fecal transplants or oral treatments with certain bacteria, may serve to aid our fight against widespread diseases such as asthma. Human microbiomes remain an emerging field of study, requiring deeper research into the microbiomes of the human lung in particular, along with targeted antibiotic treatments. Microbiomes must be understood to understand their consequence to asthma and other pathologies.